The second point is increased gluconeogenesis. Gluco substrates for gluconeogenesis is glucogenic amino acids, lactate from muscles, and glycerol from adipose tissue. And this gluconeogenesis is favored by the activation of fructose 1,6 bisphosphatase, and which is activated due to the decreased availability of its inhibitor, that is fructose 2,6 bisphosphate, because glucose level is low. And the second is induction of phosphoenol phosphate carboxy kinase by glucagon, which is the enzyme for gluconeogenesis. And this gluconeogenesis begins within 4 to 6 hours after the last meal and fully active when the hepatic glycogen store is depleted. Again, glucose 6 phosphate is the key point. Glucose converted to glucose 6 phosphate, and which takes the three pathway: glycogen synthesis, glycogenesis, and glycolysis, and the pentose phosphate pathway. And it links the pentose phosphate pathway, glycogenesis, and glycogenolysis to glycolysis. So you see the carbohydrate and lipid metabolism are interconnected. The fasting state, amino acids, glycerol, lactate used for gluconeogenesis and ketone formation, and the starvation state, fatty acids used to greater extent. Glycerol is major glucose source, and ketosis after oxaloacetate depletion. Now the fat metabolism, increased fatty acid oxidation, major source of energy, decreased malonyl coenzyme A due to phosphorylation, that is inactivation of acetyl coenzyme A carboxylase by AMP kinase. This malonyl coenzyme A is decreased due to the decreased fatty acid synthesis, because the ACC is the enzyme for fatty acid synthesis pathway. And when the malonyl coenzyme A is reduced, the inhibition, because it is the inhibitor of CPT1, that is carnitine palmitoyl transferase 1, that inhibition is removed and beta oxidation goes on uninhibited, and which generates acetyl coenzyme A, NADH, and FADH2. You have read. Now this NADH inhibits the TCA cycle. Oxaloacetate is converted to malate, so acetyl coenzyme is not entering TCA cycle, and now it diverted for ketogenesis. And this acetyl coenzyme A, which is allosteric activator of pyruvate carboxylase and allosteric inhibitor of pyruvate dehydrogenase, and that way it favors the use of pyruvate in gluconeogenesis. This pyruvate carboxylase is the enzyme for the gluconeogenesis, and PDH is for the TCA cycle. The oxidation of NADH and FADH2 coupled with oxidative phosphorylation supplies energy required for PC, that is pyruvate carboxylase, and phosphoenol pyruvate carboxy kinase reactions of gluconeogenesis. The second is increased ketogenesis in fat metabolism. These are beta hydroxybutyrate and acetoacetate. Acetone is volatile substance and it is not used for ketogenesis, not used for energy production. And these are synthesized in liver and released and used for fuel by peripheral tissues. But these ketone bodies are not used by liver because it lacks the enzyme thiophorase, which is the enzyme for ketolysis. So ketogenesis starts from the first day of fasting, and when the concentration of acetyl coenzyme A from fatty acid oxidation increases the oxidative capacity of the TCA cycle. And this coenzyme A released by ketogenesis, you have read during ketogenesis and ketolysis, coenzyme A is released there and which is utilized by fatty acid oxidation, and it continues. The brain cells uses ketone bodies if the level of ketone bodies are such high in concentration, and the need for gluconeogenesis from amino acid carbon skeletons is spared because the proteins are utilized for other vital functions. If ketone bodies are high, then it leads to ketoacidosis. Here, ketone bodies supplies 2/3 of energy to the brain, and glucose from gluconeogenesis provides the 1/3 of energy. And this is the cycle what happens from adipose tissue, fatty acids, glycerol. RBC uses glucose, produces lactate and pyruvate, and alanine from muscle proteolysis goes and utilized for gluconeogenesis. And then this ketone bodies are excreted through urine and produces ketoacidosis. Adipose tissues in fasting, carbohydrate metabolism, glucose transport by insulin sensitive GLUT4, and its metabolism is decreased because there is decreased secretion of insulin, and that leads to decreased synthesis of TAG in adipose tissue. What about fat metabolism? There is increased degradation of fat, that is pKa mediated phosphorylation and activation of hormone sensitive lipase, and that leads to hydrolyze the fat, that is TAG, and this is due to increased epinephrine and norepinephrine. Increased fatty acid release, TAG hydrolysis is increased, which is broken down in glycerol and free fatty acids. Glycerol is utilized by liver for gluconeogenesis by the enzyme glycerol kinase, and fatty acids bounds to albumin in the blood, goes to peripheral tissues where it is oxidized and produces energy, or it goes to TCA cycle for energy, or it re-esterified to glycerol 3-phosphate and generates TAG and reduces the plasma fatty acid concentration. And the third is decreased fatty acid uptake. In fasting state, LPL, that is lipoprotein lipase activity in adipose tissue is low. So, fatty acid uptake by adipose tissue is reduced. Here in adipocyte, what happens? Triacylglycerol is degraded in fatty acids and glycerol, and it goes into the blood and utilized for gluconeogenesis, and fatty acids synthesizes beta oxidation acetyl coenzyme, which goes to TCA cycle and produces energy. Here you can see in detail that glucose from the liver and VLDL from the liver comes to adipocytes, and glucose and fatty acids this produces glycerol 3-phosphate and fatty acyl coenzyme, and triacylglycerol is synthesized, and then again hormone sensitive lipase degraded them into glycerol and fatty acids. Resting skeletal muscle in fasting, resting muscles use fatty acid instead of glucose, but exercising muscles use initially creatine, phosphate and glycogen store. Intense exercising muscles, what happens? Glycogenolysis, glucose 6-phosphate, and that is in anaerobic glycolysis, lactate is produced and it goes to liver for gluconeogenesis, and this is the Cori cycle. Here is the diagram for Cori cycle. You see glucose from the liver comes to muscle, converted to pyruvate, gives you ATP, and then anaerobic glycolysis, lactate is produced, which goes to again to liver and converted to pyruvate and produces through gluconeogenesis glucose. And this is again the